Hi everyone, I'm Tehilla Fogel from Hadassah Hospital in Jerusalem, Israel, and I'm presenting you my research on cell therapy with adult human mesenchymal stromal cells for treating diabetes. So we have a rat model that demonstrates a phenomena that happens in diabetic humans in which the pancreas mass is reduced and it becomes all fatty, and we use our placental cells for treating that. So we previously reported in different studies that the treatment with adult human mesenchymal stromal cells isolated and expanded from a fetal human placenta exert pro-regenerative effects under different degenerative conditions. In the current study, we used a nutritionally induced type 2 diabetes rat model, the coin diabetic sensitive. When fed high sucrose, low copper diet, it becomes hypoglycemic. It's featured by impaired glucose-stimulated insulin secretion, pancreatic damage, loss of the acinar cells, and infiltration of adipose tissue, and activated macrophages releasing IL-1 beta. When they're fed regular diet, they maintain normal glycemic. So we investigate whether the treatment with remotely delivered placental cells can indirectly prevent or reverse the pancreatic damage, yielding a better glycemic control. So we used the CDS rats fed with high sucrose diet or regular diet as control, and we injected them IP with 5 million cells. So these are the cells isolated from the placental tissue, and they are positive to mesenchymal markers, and we isolate them from a male fetus, a placenta of a male fetus, and therefore we can validate them by XY centromere. We performed 2 sets of experiments, and the first one we did an early treatment before the pancreatic damage was evident, and the second one we did a late treatment after the pancreatic damage was clearly evident. In both sets of experiments we did 3 injections, and at the day of termination we performed an oral glucose tolerance test, and we sampled the glucose and insulin levels, and the pancreas was excised for further investigations. So to the amazing results that we got, this is a treated pancreas. You can see it fully restored its original size, like the naive control, like the healthy ones, compared to the non-treated ones in which the pancreas reduced its size, and that happened in both early and late treatments. And if you look at the objective measurement, the red bars are the treated ones. You can see that the pancreas fully restored its length and weight in both early and late treatment. So we further performed immunohistology to IL-1 beta and to macrophages. And also over here, the treated rats showed, in the red bars, clearly reduced levels of macrophage infiltration into the pancreas compared to the non-treated ones. And that also happened in both early and late treatment. So the mechanism in which these cells work, we do not know, but we assume that it has something to do with the mitochondria. So we checked that by transmission electron microscopy. And as you can see, the mitochondria from a treated pancreas preserved its original structure compared to the non-treated ones. So after we checked the pancreas itself, we wanted to check the glycemic control. And also over here, we saw that the treated rats, the red one, reduced the levels of glucose compared to the non-treated ones, and that was associated with the increase in glucose-stimulated insulin secretion. So we propose a mechanism on how that happens. So in regular inflammation, there's a lot of macrophages releasing IL-1 beta. The IL-1 beta goes into the beta cells, and it increases the levels of nitric oxide that competes with the respiratory chain over the free flow of electrons in the cells and does not allow it to produce enough ATP to secrete insulin. When we reduce the inflammation, we reduce the macrophages, reduce IL-1 beta, and therefore allow more insulin secretion. So to conclude, in both early and late cell treatment, we managed to fully restore the original size of the pancreas, to reduce macrophage infiltration, and to preserve the mitochondrial structure. We managed to reduce the levels of glucose and to increase the levels of insulin, and therefore our study suggests that the cell therapy with the placental cells has a high pro-regenerative potential for treating diabetes.